Now we're going to spend a couple of minutes reviewing the nervous system. Just like before, brief review does not substitute for being in lecture, but helpful in understanding the big picture. The nervous system is for homeostasis. At the beginning of the semester, we looked at the fact that the nervous system accomplishes homeostasis quickly, on the order of less than seconds. And even back then, we used a similar diagram. The nervous system requires sensory input by action potentials on neurons into the central nervous system for integration. Once integration occurs, then action potentials travel out to effectors on the motor. So sensory input is in, that's afferent, to the integration center, which will be brain or spinal cord. And then integration center sends information out along efferent pathways, which are the motor neurons, to the effector. It's highly organized. So that brings us to the fact that there are different nervous systems at play here. We have a brain and a spinal cord, which are the central nervous system. And then we have spinal nerves and cranial nerves that are the peripheral nervous system. These would be those sensory and motor, afferent and efferent neurons we just looked at. In the peripheral nervous system, if you divide it into its two divisions, we indeed have the afferent or the sensory neurons, and we have a motor division or efferent neurons. Furthermore, we can subdivide the motor category based on effector type. The somatic motor division is voluntary control. It's one motor neuron from the central nervous system to the effector, and the effector is always skeletal muscles. The other half, anything else in the body that's not a skeletal muscle, falls into the category of autonomic. Autonomic motor division is involuntary. This would be smooth muscle, cardiac muscle, and that means you're controlling glands, organs, blood vessels, things like that. Furthermore, in the autonomic division there are two subdivisions. The parasympathetic is in charge of what your book calls housekeeping. Parasympathetic kicks in when people are calm. It stimulates the digestive tract, it stimulates the immune system. It's all about taking care of the day-to-day things when you're in a state of calm. Its counterpart is the sympathetic nervous system. The sympathetic nervous system is there to take care of you under times of stress. It shuts down the digestive system, but it stimulates your heart rate and your breathing rate. Both of these subdivisions of the autonomic nervous system maintain homeostasis. One of them does it when you're not under stress. The other takes care of you during times of stress. They both work together. After we've looked at brain and spinal cord and cranial nerves and spinal nerves, we will come to the autonomic division. For now, you need to understand that autonomic has two subdivisions, sympathetic and parasympathetic. It is involuntary control, and it is anything in the body that is not a skeletal muscle. If the effector is a skeletal muscle, that's somatic. It's voluntary control. Somatic employs one motor neuron. And by the way, autonomic employs two motor neurons. So they're not only functionally different, they are physically different. They even have different neurotransmitters. Then we started getting into the nitty-gritty of nervous system tissue. There were two populations of cells. There's neurons and neuroglia. Neurons are the ones that are excitable. They can generate electrical events, and those electrical events fall into two categories. If the electrical event is up on the soma or the dendrites, or the peripheral process. And that electrical event does not travel. It is a moment in time. That is a graded potential. Graded potentials tend to use mechanically or chemically gated channels. If the electrical event begins on the axon hillock, it uses voltage-gated channels and it travels down the axon to the end. That's an action potential. And again, those are the roles of neurons. Now, because neurons are so special, because they're electrically active, their needs are great. So they need this other population of cells to support them, surround them, connect them, prevent them from touching each other, and to help them maintain homeostasis by regulating their environment. That's the role of neuroglia. In the central nervous system, there are 4 populations of neuroglia. We had astrocytes, microglia, ependymals, and oligodendrocytes. Astrocytes have the biggest job. They are the ones that are wrapped around neurons and capillaries. So they control the interstitial fluid here. They pump out excess neurotransmitters and ions. They control permeability of blood vessels. They physically support neurons and insulate them against touching each other. They help neurons make new synapses, new connections with each other. Astrocytes have a huge role to play. The population responsible for phagocytizing debris, or anything that does not belong, are the microglia. So microglia are kind of like macrophage for the central nervous system. And that's their sole role. Their function is to maintain health by phagocytizing what doesn't belong. Then there are the ependymals. Ependymal cells line fluid-filled cavities within the central nervous system. The fluid inside those cavities would be cerebrospinal fluid. It is the transport system for the central nervous system. Ependymal cells are ciliated, and their job is to filter and secrete and circulate the cerebrospinal fluid. Lastly, we had oligodendrocytes. Oligodendrocytes actually come from a subpopulation of ependymal cells. Oligodendrocytes are responsible for forming the myelin sheath in the central nervous system. They form it differently than the myelin sheath in the peripheral nervous system. In that, we still have the sheath, we still have nodes of Ranvier for action potentials. But there's no neurolemma. If you lose an oligodendrocyte, you lose that myelin sheath and you are not getting it back. Then in the peripheral nervous system, we had 2 populations of neuroglia. There are satellite cells and Schwann cells. Satellite cells have unknown function. They're wrapped around the soma, especially of the unipolar neurons. Schwann cells form the myelin sheath. They are wrapped around the axons, and they form it differently than the oligodendrocytes do. Their job is, again, to form the myelin sheath, but when they do so, they wrap around the axon. I'm outlining the axon of the neuron. All the rest of that is myelin sheath. It is cell membrane, just like in the oligodendrocyte, without proteins, so it's a non-polar membrane. It insulates the neuron so that wherever you have myelin sheath, you cannot have an action potential. The action potentials have to take place in the spaces between the cells, which are the nodes of Ranvier. When a Schwann cell forms a myelin sheath, it's an individual cell wrapping on an individual axon. One axon will have hundreds and thousands of Schwann cells around it. The Schwann cells wrap in a way so that the surface structure here is their cytoplasm and all their organelles. You can see the red nucleus there. That is the neurolemma. And if you damaged myelin sheath in the PNS, you can, to an extent, regenerate it. The last thing on this slide is to point out that that should say PNS. Again, serves me right for doing these things at 7 o'clock in the morning. So those are the neuroglia. Then we switched our focus to the neurons and talked about their body parts. Neurons have a cell body called a soma, or cell body, or perikaryon. In this type of neuron, which is a motor neuron, you can see that there are processes coming off the soma that branch and taper. If you have processes coming off the soma that branch and taper, those are dendrites. Dendrites receive electrical signals and bring them toward the soma, more or less. Furthermore, in a dendrite, you would have a graded potential. If you go inside the soma, you'll see that just like every other cell, except red blood cells, there are nuclei, and inside the nucleus is a nucleolus, an area rich in RNA. Neurons have specialized rough endoplasmic reticulum called nisal bodies. And if you go to where the axon leaves the soma, that area is called the axon hillock. That is where the action potential is generated. The action potential is then propagated down the axon. The axon is the long process coming off the soma that does not taper. It branches only at the very end. The exception to that rule is rare. There are neurons whose axons branch, but they're very rare. They're called collaterals, and we'll talk about them again later. Once you get to the very end, you can see that the axon branches. At that spot, that's called the telodendria. Those are the little branched endings. And the very, very, very ending is called an axon terminal. That's where the neurotransmitter gets released. You can see that in this neuron, this motor neuron, it is myelinated because there are Schwann cells wrapped around it. And between the Schwann cells, we have a little bit of what I call naked neuron, and that is where the action potentials take place. Remember, ions are not permeable across a lipid layer, so the ions can't move across the myelin sheath. They can only move where you have a naked neuron, and that's what an action potential is. So you should take a second, you should pause here, you should review what we've talked about so far. We looked at the divisions and the subdivisions of the nervous systems. We focused on neurons versus neuroglia. You should remind yourself now what all the neuroglia are, what their functions are, and then you can focus on this slide and remind yourself about the neurons and all their different parts. Now that you've reviewed, hopefully, the other things about neurons is that they're very long-lived. They're amitotic, which means they don't divide. They physically can't. They have a very high metabolic rate, which is why you get so tired when you study all the time. Remember, their job is electrical signaling, and they can also do cell-to-cell signaling at their terminals. Alright, so again, a little bit of a review. Remember, the soma is where you find the nucleus, the nucleolus, the nisal bodies, and the axon hillock. Dendrites are tapered and branched. They are the receptive ends. They receive signals, and they generally do graded potentials. Then we have axons. Collaterals or branched are rare. The axons generate and propagate action potentials. If you have a bundle of myelinated axons in the central nervous system, we call it a tract. If you have a bundle of axons in the peripheral nervous system, we call it a nerve. We classify neurons in part based on shape. How many processes come off the soma? If more than 2 come off the soma, those are multipolars, and they're on the left. There are a lot of different kinds of multipolar neurons. They're the major type of neuron in the central nervous system. They're the most abundant ones in the body. And again, you have a lot of different variations of multipolars. If there are exactly two processes coming off the cell body, that's a bipolar. Bipolars are fairly rare. They're found in specific sensory organs, the eye, the ear, the olfactory mucosa, but they're pretty rare except in those spots. Then lastly, we found the unipolars, one process coming off the soma. Those are the sensory neurons, so they're primarily peripheral nervous system. Their cell bodies are in a specific place of the spinal cord called the dorsal root ganglion. Okay, so we looked in class at how neurons transmit these electrical signals. And we did it a few times. It's a critically important concept. It's how neurons function. If you block this function in any way, you interfere with the neuron's ability to transmit information through the body. So in order to do these electrical events, the first thing any neuron has to do is it has to create a resting membrane potential. It has to pump ions in specific locations and create a difference in charge, electrical charge, across its membrane. Then, when the time is right, the neuron will open gates in its membrane, which allows ions to move. When ions move, that means charge is moving. And if you can move charge, you generate electricity. The third step is to reestablish their resting potential, to close the gates back up again and pump the ions in the correct places. So again, step one, create a resting membrane potential by having closed gates and pumping ions. Then open and close gates strategically to let ions flow to generate electricity. Then finally, close the gates and pump the ions back into their original position. So how does a neuron do this? When a neuron goes to create a resting membrane potential, realize that there's things stuck in that neuron's axolemma. So the green here represents a little piece of the axon, and the line at the edge of the green is my axolemma, my cell membrane. Inside the cell membrane is a ion pump, a protein pump that uses ATP to move ions. We also have gates, a gate for sodium and a gate for potassium that are going to open and close strategically. And combine that with the fact that inside the cell are giant proteins and other molecules that are relatively negatively charged that can never exit the cell anyway. So it turns out this pump moves 3 sodium ions out and then 2 potassium ions in. At the beginning of the story, the sodium voltage-gated channel is closed, so while sodium can leak across the membrane, it's not moving in large amounts. Also at the beginning, potassium's voltage-gated channel is closed as well. And again, potassium leaks across the membrane, but it's not moving in large amounts, and both ions, even though they might leak, are pumped right back to their original position. So at rest, we have this situation where I have more positive outside the cell than inside the cell. The inside the cell is relatively negatively charged because of the ions that are in there and because of the fact that I moved only 2 pluses in for every 3 pluses that I moved out. So you can see charge is not balanced. It's negative inside and positive outside. So in this position, can sodium cross the membrane? No, because its voltage-gated channel is closed. It won't cross the membrane in large amounts. It does leak. It's the leakage of the ions that keeps this membrane excitable. Is there a sodium gradient? Yes. There's lots of sodium outside, but little sodium inside. So if I allow sodium to move in large amounts, sodium wants to go into the cell and follow its gradient. Right now, can potassium cross the membrane? Not in large amounts. Again, its gate is closed, and so it's leaking, which keeps the membrane excitable, but it's being pumped right back out. Is there a potassium gradient? Absolutely. There's a lot of potassium inside the cell, but very little outside. So at rest, if I were to measure this electrical potential, it would measure out as negative 70 millivolts. That is the resting membrane potential. So this membrane is sitting here at minus 70 millivolts. The protein pump is constantly working. The ions are constantly leaking, but they're being pushed back by the pump. If a trigger happens, some stimulus somewhere happens, so that sodium specifically begins to move across the membrane in more amounts, it's leaking a little bit more. Will that affect the membrane potential? Yes, because sodium is positively charged. So as more and more and more positive enters this cell, that's going to change the charge. If enough sodium leaks into the cell such that the potential goes from negative 70 up to minus 55 millivolts, magic happens. At minus 55 millivolts, we have reached threshold. At threshold, sodium's voltage-gated channel opens. If sodium's voltage-gated channel opens, can sodium move across the membrane in large amounts? The answer to that is yes. Well, the next question is, what direction? Sodium wants to follow its gradient. And even though some has leaked, not that much leaked. So once you open the gate, sodium flows into the cell in very large amounts. Sodium is positively charged, so will that affect the membrane's potential? Absolutely. You're now adding even more positive to the inside of this cell. So this thing's going to go from minus 55 and get more and more positive until you reach the point where you have a plus 30 millivolt potential across this membrane. At plus 30 millivolts, magic happens again. At plus 30 millivolts, sodium's gate closes and potassium's gates open. Now, can potassium move across the membrane? The answer, in large amounts. The answer to that is yes. What direction? Well, potassium will follow its gradient, and its gradient is out. Will that affect the membrane potential? Absolutely, because that now means you have lots and lots of positive charge leaving the cell. This cell is going to go from being plus 30 millivolts on the inside back down to the negative millivolt range because all that plus has left. It repolarizes and hyperpolarizes until it gets way below minus 70, down to minus 90, minus 100. That's why it says minus 90-ish. Eventually, potassium's voltage-gated channels will close. And when they finally do close, that membrane will stop becoming more and more negative. And because both gates are closed, but the pump's been working the entire time, it just couldn't keep up, we eventually get back to our resting membrane potential. So we've done a little cycle. We're back to where we started. Then we talked about how an action potential cannot cross a physical space. I can do that action potential. I can start it in the axon hillock. Let me go back. All right, I can start it in the axon hillock. It is generated because I had enough graded potentials occurring on the soma and dendrites to cause that sodium to leak across the axon hillock. And I hit threshold at the hillock. Once I hit threshold at the hillock, when an action potential is taking place at the membrane of the axon hillock, that causes the membrane at the next node downstream to leak sodium, and it will hit threshold, which causes the membrane at the next node downstream to leak sodium to threshold, which causes blah, 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 blah. You get the point. Once an action potential starts at the axon hillock, it self-propagates to the end. So you just really have to hit threshold at the axon hillock. The rest takes care of itself. When we looked at action potentials, we said they are all or none. You either get it in its entirety or you don't have one at all, because it depends on hitting threshold. We said the term saltatory conduction. That means that it hops on the nodes of Ranvier because it physically can't occur in myelin sheath. But that's okay. That makes the transmission happen even faster from beginning to end. But we also said that an action potential requires membrane-bound objects. You have to have a pump, you have to have gates, you have to have some wall so that you create ion gradients. Okay. That means that an action potential cannot take place in an empty space. And unfortunately, because neurons are so electrically active and so sensitive, we can't let them touch. So when you get to the axon terminal, shown in yellow here, of the first neuron, and the soma or the dendrite or the axon hillock of the second neuron, how are we going to get the action potential from the first neuron in yellow to the second neuron in gray if they can't physically touch and the action potential cannot take place on the membrane? Well, the solution is transduction. That means we're going to change how we do this in the cleft. So here's how the cleft works. It's a transfer of information. An action potential comes down from the axon hillock all the way down the axon through the telodendria to this axon terminal. When the action potential hits the axon terminal, that voltage change opens calcium's voltage-gated channels. Calcium flows into the membrane, and that calcium triggers exocytosis of these little vesicles here. Those vesicles contain neurotransmitter, a chemical. So when they accomplish exocytosis, you are now releasing neurotransmitter into this space. Neurotransmitter diffuses across the cleft to its receptors on this postsynaptic membrane, neuron B. If they can bind to their receptor, that receptor will open and allow ions to flow. Those receptors are a kind of voltage-gated channel. If you look at the diagrams over here to the right, you can see that when the neurotransmitter binds, that these things open to allow ion movement. If you pull the neurotransmitter off, these things close and you no longer have ion movement. So now I say again, pause the slide, or go backwards. Review action potentials in all their stages, from polarization, depolarization, repolarization, hyperpolarization, and then back to rest. Review the events of the synapse. Action potential opens calcium's voltage-gated channel. You get exocytosis of neurotransmitter. Neurotransmitter fuses with its receptors, which opens and allows ion movement. Only after you have these two processes down should you move to the next slide. Hopefully you took my advice. Now, because neurotransmitters affect the second neuron, you cannot leave neurotransmitter sitting there forever. So there are 3 ways we get rid of neurotransmitter once we've dumped it out. One is reabsorption, the other is with enzymes, and the third is diffusion. one of the enzymes is acetylcholinesterase. ACE at the end means enzyme. This enzyme degrades acetylcholine. Acetylcholine in the somatic division is excitatory. Your motor neurons dump neurotransmitter called acetylcholine onto your skeletal muscle cells, and that triggers them to contract. So if you blocked acetylcholine, or a mad scientist created a chemical that acted like acetylcholinesterase, you would prevent someone's skeletal muscles from contracting. In the world of reabsorption, we have a couple of options. The presynaptic axon terminal can reuptake. It has pumps, and it can pull neurotransmitter out of the cleft and reabsorb it. In the central nervous system, astrocytes also do the same thing. And lastly, that neurotransmitter can simply diffuse away from the synaptic cleft, and if it's in the central nervous system, an astrocyte somewhere will pick it back up again. Now, not all neurotransmitters are created equal. Some exhibit an excitatory postsynaptic potential. If it excites, that brings the neuron closer to threshold so that it is more likely to fire an action potential. The laws of summation, which we'll review in a second, apply. If a neurotransmitter hyperpolarizes a membrane, then it is an inhibitory postsynaptic potential. An inhibitory postsynaptic potential brings the second neuron further away from threshold. It hyperpolarizes it by moving chloride or potassium. That means an action potential is less likely to occur, so we've blocked the second neuron. And again, the rules of summation apply. So what's summation? Summation in Miss Klein language means enough. You have to get enough neurotransmitter to cause an effect. Well, there's two ways I can get enough neurotransmitter to cause an effect. I can either dump a lot of neurotransmitter, I can do a lot of exocytosis at one moment in time, or I can release neurotransmitter over a period of time. If I dump a lot of neurotransmitter at once, one moment in time, lots of terminals at once, that's called spatial summation. If I dump neurotransmitter over a period of time, that's temporal summation. Either way, if I can accomplish spatial summation or temporal summation, I will hit threshold at the second neuron or successfully inhibit that second neuron. So now, that comes to the end of our review. You should use your objectives for this chapter. Go over those objectives, use the lecture notes, the PowerPoints in your textbook, so that you have an in-depth, full understanding of every objective. Once you accomplish that, then you want to take a lot of practice tests repeatedly. Don't just take one practice test once. Do it again and again and again. As you take practice tests multiple times, that is studying. It also cements that information in your brain. You're less likely to forget it, and you're more likely to stay calm during the exam. You have practice tests on Blackboard, you have them with Professor Hubley, Professor Imholtz has more. If you have Mastering AMP Code, there's some in there. If you have the old textbook website, there's some in there. There's practice tests all over the place. And you know, when all else fails, use Google. Finally, when you do go in to take this next test, make sure, please, 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 that you use your test-taking strategies. Imagine me sitting next to you and coaching you through it. Hear my horrifying, obnoxious voice in your brain telling you, read the question carefully. Do not look at the answer options. What are the key words and what's this question asking you? What's everything you know? Only after you've done that should you go to the answer options. And remember, read them carefully. Every answer option needs to convince you that it's the correct one. Get a good night's sleep to the best of your ability before this next test. Eat something healthy and long-lasting to the best of your ability before this next test. And please don't hesitate to ask questions or see the campus tutors or see me. That's it for the review.